Okay, everybody, welcome back. We're going to begin today just with the last little details about protecting groups, or rather the first protecting group that you're going to encounter in this class. The idea is that sometimes you're using reagents that have characteristics that might interfere with other functional groups in the molecule, such as the organolithium or the Grignard or the cuprate, which, though they're good nucleophiles, are also pretty basic, and so you can't use protic solvents. And if you have a molecule that has an acidic proton in it, the Grignard reagent or the organolithium or the cuprate is just going to deprotonate the alcohol, and then you don't get your desired product. So what we do is, to prevent that undesired functional group from causing trouble, we get rid of it with a protection reaction. It's got to be easy to install, it's got to lack the acidic proton, and then it's got to be easy to remove. So this is the sidestep strategy. Protect the alcohol, do the nucleophilic addition to the carbonyl, then deprotect the alcohol. None of you have ever seen the Dick Van Dyke Show from like the 60s, but if you ever do, there's an opening sequence, and you never knew whether, as Dick Van Dyke rocks over to kiss Mary Tyler Moore, whether he's going to trip over the ottoman or sidestep it. Well, this is the sidestep at that moment. Just Google it. It's on YouTube. You should be able to see it. It's classic. Anyway, let's introduce that protecting group. The protecting group of choice in this chapter for alcohols is the silyl ether. Silyl means silicon-based ether. An ether is an oxygen bonded to two carbons. A silyl ether is an oxygen bonded to a carbon on one side and a silicon on the other side. And silicon's similar to carbon, though it has some different properties. It's one row down, same column of the periodic table from carbon. So here is an example of a silyl ether. I'll draw it on the molecule that we were interested in. The silyl ether would have a silicon bonded to that oxygen, and then there are usually other alkyl groups attached to the silicon. This is the one that's used in your text. It's tert-butyl dimethyl silyl ether. Okay, and of course we get tired of writing all of that down, so we, like alphabet soup in organic chemistry, tert-butyl dimethyl silyl ether, we're going to abbreviate as the TBDMS group. And in fact, most organic chemists are going to get so tired of drawing silyl ethers that they will just abbreviate it like this. And that's what your text does. Okay? So that's the protecting group. You can see that it lacks the acidic proton. So we check that particular box. We got two other boxes to check. One is it easy to install? The answer is yes. So let's show you how to install the silyl ether group, the protection reaction. You start with your alcohol, and then the reagent of choice is the same silicon bonded to the same three alkyl groups, but on the other side is chloro, which is a leaving group, okay? And we won't worry about the mechanism of this reaction. Silicon being larger than carbon, the rules for substitution reactions on silicon are different than they are for carbon. We won't worry about this mechanism at all. Essentially, though, it's a substitution reaction. We're going to use the OH as a nucleophile, and then we will attack the silicon, and the chloro leaving group's going to leave. Now, we're also going to need a base, so TBDMS chloride, or tert-butyl dimethyl silyl chloride, is the electrophile. This is TBDMSCl is how we'll abbreviate it. We're also going to need a base, and the purpose of the base is just to take care of the proton on the alcohol, because in the tert-butyl dimethyl silyl ether, the proton is gone. The base of choice is a very mild non-nucleophilic base. It's called imidazole. It looks like this. It's a 5-membered ring containing nitrogen, and the basic nitrogen is right there, lone pairs on that nitrogen. We'll encounter imidazole again in our chapter on aromaticity, when we'll have a little bit more to say about why it's basic. But those are the conditions, and you do those, put those two reagents together with your alcohol, and very fast, and in basically 100% yield, you get your tert-butyl dimethyl ether. So that's the protection reaction. That one, again, we're not doing mechanism. You're probably going to have to memorize the details on that. And you're going to get confused a little bit with alphabet soup. What does TBDMSCl even mean? So practice, use potentially the flashcard method if that helps you. Then we need to do the deprotection reaction. The deprotection reaction is also a substitution reaction, but it relies on the fact that silicon-fluorine bonds are more stable than silicon-oxygen bonds. Let's just, for the sake of our argument, go ahead and in between the protection and deprotection steps, let's do the desired nucleophilic addition reaction, where we react our phenyllithium with our alcohol, with our ketone, and then after aqueous workup, we've made the correct carbon-carbon bond, and the alcohol didn't interfere with things because of that silyl ether. Then the deprotection reaction goes like this. It relies on the fact that a silicon-fluorine bond is more stable than a silicon-oxygen bond. So you need a reagent that uses fluoride as a nucleophile. We could say, we won't worry again all in detail about the mechanism of this reaction. Essentially, it's a substitution reaction. You need a source of fluoride that is soluble in organic solvent, and so one of the things they do is they use as a counter ion a positively charged ion that's got a lot of carbons in it. This is a quaternary ammonium group. It's called tetrabutyl ammonium, and the butyl groups make it soluble in organic solvent, and so the solubility of the positively charged ion helps the fluoride get into organic solvent, and forming the new carbon fluoride bond releases the silyl ether. I'm sorry, in the deprotection reaction, this should be the alcohol, okay? This is abbreviated as because we like acronyms in organic chemistry. We don't like them nearly as much as they like them in biochemistry, but TBAF, tetrabutyl ammonium fluoride, is the reagent that does the deprotection. So you'll have a chance in some practice problems to work on that. Be sure you know how to use the protection, deprotection strategy. Questions about that? Okay, yes?
Where does it get the proton to become alcohol?
Where does it get the proton to become the alcohol again? Probably from aqueous workup. Yep, good. Others? Okay, so we now need to move on to chapter 14. The theme in chapter 14 is going to be carbonyl chemistry reactivity based on just looking at mostly aldehydes and ketones. And then we're going to be looking at nucleophiles that are not as strong of a nucleophile as is, say, the Grignard reagent or the organolithium reagent. So we'll start probably with the most complicated stuff. It's probably not where your chapter starts, but we'll go ahead and dive in. The idea here is, what do you do if you have an aldehyde or a ketone and you want to react it with an amine as a nucleophile? Now, what I'm going to show you will make sense based on our discussion so far, though it is a little bit different from the way your text shows things. Your text will show a mechanism here for this reaction that I'm introducing that is perhaps a little simpler but less realistic. Sometimes in science, as we teach you new concepts, we oversimplify to you. This is a case where I feel like it's useful enough for you to know what's actually going on that we're going to talk about it, okay? So if you have a carbonyl compound. Here's the carbonyl carbon, here's the carbonyl oxygen. The pi star is the LUMO. You expect the nucleophile to attack the carbonyl carbon, but the question we've been asking is the nucleophile good enough to attack the carbonyl carbon directly to make a negatively charged intermediate? We could go ahead and draw what that intermediate would be. I'll even draw the arrows that are the instructions for that intermediate so we can ask the question, is this reasonable? If we make a new bond between carbon and nitrogen, that nitrogen's already bonded to three things, so it would have a positive formal charge. And the question we gotta ask is the nucleophile a good enough nucleophile to make a negatively charged oxygen intermediate? Now, the way I've showed you to think about that is you ask, okay, how basic is this amine? If you want a sense for how basic or reactive the amine is, you can look at its conjugate acid, which would be the ammonium ion, and then look at that pKa, which is around 11. And then if you want to compare that to the reactivity of the negatively charged oxygen, you think about the conjugate acid of that negatively charged oxygen and its pKa, which is 16, and you would conclude that maybe I'm a little bit worried about that. Maybe it's problematic for this neutral amine, which is much less reactive than a negatively charged oxygen, to make that intermediate. And in fact, that's true, and this is a situation where we would say probably not reasonable, and we probably need acid catalysis. Remember, acid catalysis is something that we do to make the nucleophilic addition to the carbonyl carbon more reasonable. So if you go look up, this reaction is called imine formation. If you go look up the mechanism for this reaction in your text, they're going to go ahead and show you this first step as though it's totally reasonable and fine. In practice, you almost always need an acid catalyst when you do this in the lab. So I'm going to show you that, and you'll just have to deal with the fact that the mechanism I'm showing you is more realistic than the one that's shown in the text. All right. So when we need acid catalysis to make imine formation occur, there are a couple of options for what might go on. You could envision, for example, the first step being the carbonyl oxygen picks up a proton from your acid catalyst to generate this oxonium ion intermediate. And then you could imagine that oxonium ion intermediate could be attacked by your nucleophile. Again, the LUMO would still be the carbon-oxygen pi star, but the presence of that positively charged oxygen makes the oxonium ion more reactive than the starting ketone or aldehyde that we began with. I don't care which it is. We'll be doing aldehyde to be consistent with what's above. Do you see how that changes things? Then our consideration is comparing the strength of our nucleophile versus the reactivity of a neutral oxygen. So if your intermediate has this neutral OH group, you can probably get away with much less reactive nucleophiles doing the attacking. The other reason to think about that oxonium ion as being way more reactive or more electrophilic is to draw the resonance structure of the oxonium ion with a positive charge on the carbonyl carbon. And that's different from, say, the resonance structure for an aldehyde that's not already protonated, because in that resonance structure, the oxygen's negatively charged. What this shows you is, in the oxonium ion, a lot of that positive charge is actually felt on that carbonyl carbon. Okay? So that's option one. We'll just be general here, acid-catalyzed nucleophilic addition. Option one is and I need a little bit of space. I apologize for the poor penmanship. Option 1 is protonate first, protonate the carbonyl oxygen first, then in a second step you do nucleophilic addition or nucleophilic attack. And that, this doesn't matter, and I'm not going to test you on this term, but you're going to encounter this term in biochemistry, so we'll talk about it now. This is called specific acid catalysis. Anytime you protonate first, and then the reaction happens, that's called specific acid catalysis. Another option, which may not be immediately obvious to you, is that you could protonate the carbonyl oxygen while the nucleophile attacks in the same step. So to show that, we would have our carbonyl compound here, we would have our nucleophile here, we would have our acid here. The nucleophile attacks the pi star, and we break the carbon-oxygen pi bond, but we never get to a negatively charged tetrahedral intermediate because as this bond is breaking and negative charge is building up on the carbonyl oxygen picks up a proton from the acid. And that will get us to an intermediate that looks like this, in which we go straight from the carbonyl and the nucleophile to the neutral tetrahedral intermediate where the oxygen is not negatively charged. Does that make sense? Yeah.
Would that still be called specific acid?
No, if you do this way, when you protonate first, while the nucleophile attacks, it is no longer called specific acid catalysis. It is called general acid catalysis. And you'll see examples of that in biochemistry. A lot of enzyme active sites do general acid catalysis. Okay, questions? Yes?
So like going up to back to option 1, why would we choose option 1 over option 2?
Why would you choose option 1 over option 2? It's a really interesting question. You could imagine two things impacting whether it goes 1 versus 2. The first consideration is how strong of a nucleophile is your nucleophile. If it's decently strong, it might not wait for protonation to happen first, but it might protonate while. Okay? If it's weaker, it might have to wait for the acid to protonate first. Second consideration is how strong is your acid. If it's a super strong acid, maybe you protonate first and the nucleophile doesn't have a chance to attack yet. We're going to see in this chapter that nucleophiles like water and alcohols prefer, because they're weak nucleophiles, for protonate first and then they'll attack. Okay, so the reactions that go by this mechanism you're going to learn are called acetal formation and maybe hydrate formation, ketone or aldehyde hydrate formation. The reactions that go by this second mechanism are going to involve slightly stronger nucleophiles, like amines. And the reactions that go by this mechanism are called imine formation reactions, and then its close sibling enamine formation. Both will proceed via this method. The nucleophile, of course, is a primary, say, or a secondary amine. Amines are more nucleophilic than alcohols. You can tell that by comparing the pKa's of their conjugate acids, or you can know it intuitively just by saying, well, nitrogen's a little less electronegative than oxygen, meaning lone pairs on a nitrogen are higher in energy than lone pairs on an oxygen. Okay? So let's go back to imine formation. We started our discussion today with, all right, let's use our amino group as a nucleophile and the aldehyde as an electrophile, and we said we needed an acid catalyst. So let's return to that and show you how that happens. We'll use the same aldehyde, And then let's use that same primary amine. And we'll use some acid catalyst. Your choices here are many. You want something, generally the criteria needs to be, it should probably be organic soluble. Good options include acetic acid, which is a carboxylic acid, or another acid that you encountered before, TSOH, para-toluene sulfonic acid. This is a basically organic soluble version of sulfuric acid. And there are others that you might include using. When you're using an acid catalyst, you don't need very much acid at all. So anyway, in problems that I write, I will include an acid catalyst. I'm not remembering whether the book will always do that or not. One particular challenge that I think you might face is if you have an amine and an aldehyde and then an acid catalyst. The acid catalyst has a carbonyl oxygen too, and there's a potential for you to get confused about what on earth you're supposed to do. For that, just focus on relative reactivity of carbonyl compounds. Remember that aldehydes are at the top of reactivity. Carboxylic acids aren't really electrophilic at the carbonyl carbon. They're acidic at the acidic proton. We'll do chemistry with carboxylic acids in chapter 16, but for now, if you see the aldehyde and the amine, and there's also a carboxylic acid present, that carboxylic acid is just the catalyst. Okay? All right, so let's show how this works. The amine attacks the carbonyl carbon at the same time as the carbon-oxygen pi bond's breaking, at the same time as the carbonyl oxygen picks up a proton from the acid. All of that's in one step. I'm using equilibrium arrows here. I know that creates anxiety among at least some of you. Do I need to know that that's an equilibrium arrow? I'm going to include it so to help you see when steps can go backwards. But because on my exams, you're going to be, on a mechanism question on my exam, I'm going to be showing you a potential mechanism step and then say, is that reasonable? Or which of these mechanism steps is reasonable? Or which of these mechanism steps is incorrect? There really aren't any cases where I'm like, should that arrow be an equilibrium arrow? It's going to help you if you can understand that and follow it along, but you're not going to face the question, should that arrow be an equilibrium arrow? Does that make sense? Okay. You don't trust me yet, you're not sure. Stay tuned, ask more questions, all right? So after that initial addition step, the intermediate you've got for imine formation looks like this. What used to be the carbonyl oxygen is now neutral, and then the nitrogen should have a positive formal charge on it. Now, at this stage, we can ask the question, this was our nucleophile, and it generated this neutral oxygen. Would it be reasonable for a neutral oxygen to kick off the amine as a leaving group? And the answer is probably. A neutral oxygen should be able to kick off a neutral nucleophile, right? So this step is reversible. Anytime a step is reversible, one of the things you worry about is how do I know whether it goes backwards or forwards? You don't. And often when reactions are reversible, you the experimentalist, have to do something to drive the reaction to completion. I will show you one of those options in a little bit, okay? Now, because this is an acid-catalyzed reaction, I should draw the conjugate base of our acid here. And ultimately, we need to regenerate our acid. And so what will happen next, if we want to, of course, the oxygen could kick back down and kick off the amine as a leaving group. That would be the backward step. But what if instead the negatively charged conjugate base removed a proton from the nitrogen? That would make the nitrogen a much worse leaving group. Do you see how before, if nitrogen left, it could leave as a neutral amine? Once we remove that proton, if nitrogen got kicked off, what would it have to be? What would be the leaving group if what's in purple got kicked off? Not that. You see how that would have to be the leaving group now? And that's actually a crazy bad leaving group. If you draw its conjugate acid, the conjugate acid of that leaving group is the neutral amine, which has a pKa of 38. Okay? So just by removing a proton on the nitrogen in that intermediate, we can turn it into a not a good leaving group. And this intermediate has a name in your text, and that name is carbanolamine. It's not the end point of this reaction, but it's an important intermediate along the way. One of the things that's sort of useful to pay attention to is how that even though we've changed a lot here, we still have the same number of carbon heteroatom bonds. Heteroatom, I mean anything that's not carbon, nitrogen, or anything that's not carbon or hydrogen. So 2 carbon oxygen bonds here, 2 carbon heteroatom bonds in the carbanolamine. We haven't changed the oxidation state of the carbon at all. We've simply added a nucleophile. All right. So the reaction doesn't stop here. Instead, lone pairs on the oxygen can attack the acid, and that will generate the following intermediate, which looks like this. And you can see that acid catalysts are really useful because they change who the good leaving group is. Do you see how before, at this stage, those folks sound like they're having more fun than you. You see how before at this stage, the nitrogen, the amine group, was still a decent leaving group, but we removed a proton and now it's not. Similarly here, OH minus is not a good leaving group, but suddenly we've protonated it to become water, which can leave as a neutral leaving group. Water is a good enough leaving group for now the nitrogen to act as the kicker-offer, and then the water leaving group leaves. So let's draw what we get after that. Here's the water leaving group, and then here's the nitrogen. This is the one we originally added as our nucleophile, and there's a positive charge on that nitrogen now. Water, the oxygen that used to be the carbonyl oxygen, is now gone. And this is actually how you push the reaction forward. If you remove the water, you can use Le Chatelier's principle to drive the reaction forward. And there's some specific techniques that you can use in the lab to do this. One of them that you will use in the 353 lab is to use some kind of setup that absorbs water. So there are some salts like magnesium sulfate or calcium chloride that are what's called hygroscopic. They absorb water, and you can use them to help drive reactions like this forward. Okay. Lastly, if you're following along about our acid, let's highlight our acid in green, because in all cartoons acids are green, even though I can't think of an acid that is green. We deprotonated the acid, then there's the conjugate base. We regenerated the acid, then we deprotonated it again, and there's the conjugate base. Our final step needs to be to regenerate the acid, and so to do that, we'll have the conjugate base of our acid remove a proton from the nitrogen, and that will give us the following product and our regenerated acid. This product looks like a carbonyl, only the carbonyl oxygen is a nitrogen, and the nitrogen is attached to something else. That functional group is called an imine, and it is sometimes called a Schiff base. We hear both of those terms and should be prepared for either of them interchangeably. Okay, questions about the imine formation mechanism? Yeah.
Going back to that third intermediate, I think it's carbonyl.
Yep.
Why would the acid, or sorry, right before that, why would that conjugate base deprotonate the nitrogen and not the oxygen, not the alcohol?
Why would the conjugate base deprotonate the nitrogen and not the alcohol? If it deprotonates the alcohol, that's the backward step. Think about the attack step. It involves nitrogen attacks carbon, break the pi bond, and oxygen removes proton. The reverse of that is, starting from this intermediate, conjugate base deprotonates oxygen, electrons kick down to form pi bond, and leaving group gets kicked off. So what you just asked is totally reasonable, and it is the backward step.
And does it happen? Because it looks like...
Yeah, it does all the time. That's what this arrow means. Yeah. Now, I haven't drawn that step for you, but we will, because it turns out that you can start with the imine, add an acid catalyst, and add water, and you can go all the way back to the aldehyde starting material. Yeah. In fact, that kind of process is really important for vision, and we'll talk about that maybe sometime. Okay? Other questions? Yes? Yeah, what happened to the hydrogen at the bottom of the amine? I just randomly decided to make it a methyl group, and yeah, sorry about that. Good catch. What else? Yeah?
Is it important to use an acid that's strong but not too strong? It seems like you got to go back and forth a lot of times. Is that why we're kind of
Yeah.
just specific acidic? Or
Yeah, that's a really fascinating question. Do you need an acid that's strong but not too strong? And especially in the case of the amine, that's an important consideration, because one of the things the acid can do instead of protonating the oxygen is just to protonate the amine to make the ammonium salt, and then it's not a good nucleophile anymore. So there's actually a trade-off. People have measured rates of imine formation based on like pH and or pK of the acid, and there's a sweet spot. You get too acidic and it's not great. You get too basic and it's not great. Okay, what else? Just for fun, let's do the backwards reaction. Now, sometimes I tell students, you should be able to do the backwards reaction, and I just say, go figure it out. And they are universally uncomfortable with that. So we're going to try something new today, and I'm just going to show it to you. Is that, yay? All right. You don't like going and figuring stuff out on your own. That's all right. That's fine. Okay. The fun thing about imine hydrolysis, you're not going to like this, even though I'm going to show it. The fun thing about imine hydrolysis is it's the microscopic reverse of imine formation. So it turns out when you draw a mechanism and a reaction is reversible like this, the intermediates for the backwards reaction are exactly the same as the intermediates for the forward reaction. So I'm going to erase all the arrows, and I'm going to erase all the annoying notes from what we did above, except it's taking too long, so I should have just left them. Yep, arrows, arrows, arrows gone. Explanatory garbage also gone. We'll still call that the carbonylamine because it's an intermediate on the pathway. We're also, whoops, keeping the highlighting but getting rid of the arrows. Okay. So, there we go. The goal in imine hydrolysis, the overall reaction, is to start with the imine. In this case, we're going to add just acid, but acid and water. So we can use any aqueous acid for this. And in the end, we're going to get our aldehyde and our amine back. And probably the amine's going to be in its protonated state because we're under acidic conditions. So let's show how that might happen, starting with the amine. So if you know what the intermediates are, figuring out the arrows is not too much of a problem. For example, I'm starting with the imine, and I'm using an acid, and I want to get to this state where the nitrogen has a positive charge. This intermediate is called an iminium ion. Okay? How do you think I get from the amine to the iminium ion? What's changed? What's the new bond? Nitrogen, hydrogen. Therefore, how should I draw my arrow? Lone pairs on the nitrogen attack the acid, right? Okay, great. And if I look at what I had before, that intermediate shows a protonated nitrogen, and it also shows the conjugate base of my acid. So we're good to go. Now, from that iminium ion, the previous intermediate in imine formation was this intermediate where the oxygen is positively charged. Here, with the iminium ion, I have the iminium ion and water. How do you think I get from here to here? What does the attacking? Water's the attacking. Lone pairs on the water attack the carbon-nitrogen pi star, breaking the carbon-nitrogen pi bond. Follow along. That should give me this exact intermediate. Okay. Now, how do I get from here to the carbonylamine? Pay attention to what's changing, what's different. We used to have a proton on a positively charged oxygen. Now we don't. We used to have a conjugate base. Now we've got the conjugate acid. That tells you unambiguously what you have to do, which is what? Yeah, the conjugate base of your acid has to steal a proton from the oxonium ion. That gets you to the carbonylamine. All right, the previous intermediate had a proton on the nitrogen. How am I going to do that? Just use the acid again. Then, and this part might be somewhat tricky, you'd have to go back and probably look at the mechanism for imine formation. I need to use the conjugate base to remove a proton from the oxygen at the same time as electrons are kicking down and kicking off the nitrogen as a leaving group. So if you'll compare what we just did to imine hydrolysis versus what we did in imine formation, all the intermediates are exactly the same. And we've used the intermediates as a clue for how to push the arrows. All right, questions about that? Any food scientists out there, nutrition, anybody? No? You ever heard of the, the Maillard or Maillard reaction? When you cook bread or other baking in the oven and you're supposed to cook it until it's nice golden brown, the coloration you're seeing on the surface of your baked goods starts with an imine formation reaction. There's a lot of other things that happen, but baked goods are full of carbohydrates, which have aldehydes in them, and they're also got some protein in them, and proteins have amines in them. And so this is a reaction that occurs between that can occur between the carbohydrates, the aldehydes of carbohydrates and the amines of proteins. Starts out with imine formation, and then there's some other downstream reactions that imines can do that generate the color and the flavor associated with like the crusts of baked goods. A couple other places that imine formation shows up in your life. If you want to monitor blood glucose levels, you can do that with various methods, and you can get an instantaneous measure of glucose. But if you want to know, say over the past month, what's my blood glucose level been on average, what's the right measurement for that? Anybody know? You heard of, what is it, have you heard of A1C for diabetes, diabetes monitoring of glucose? Okay, that stands for, HB stands for hemoglobin, and A1C stands for like a modified form of hemoglobin. Here is the blob of hemoglobin. Well, I'm not going to draw out its three-dimensional structure because you don't want me to. But you should know that hemoglobin has amino groups on it. This is something that you will learn is the side chain lysine, which is an amino acid, and it has a primary amine group on it. And then you may have seen before the structure of glucose, which we'll talk about very soon. Glucose looks like this. But glucose has an open chain form that's not like that. It's actually an aldehyde. We're thinking ahead, and I'll tell you only at this stage that you get this cyclic form of glucose based on nucleophilic attack of this OH group on this aldehyde. You can probably figure that out at this stage, how that happens. But that aldehyde is reactive, and it can modify via imine formation some of the lysines of hemoglobin to generate this kind of intermediate where R is the rest of glucose. That's called glycated hemoglobin. And you can see if you could sense how much hemoglobin you have that's glycated, that would provide a record of how much glucose has been in your blood. The more glucose is in your blood, the more of it will be in its aldehyde form, the more glycated hemoglobin you're going to have. And I think red blood cells turn over on the rate of maybe once a month or so, every 30 days or so. So how much glycation or imine formation happens to hemoglobin is a pretty good analytical indicator about how much glucose you've had in your blood over the past few days. So they will monitor A1C hemoglobin over time to see whether you might be pre-diabetic or otherwise. You heard that before, sort of? If you do now, you've got an idea. The other place that imine formation will show up is in vision. How much do I want to say about this? What the heck. You want to know how you see, right? Okay, so at the back of your eyes, you can Google this if you want a pretty picture of it. You have retina. In the retina is rod and cone cells. Embedded in the rod and cone cells is a membrane protein called rhodopsin. Rhodopsin, here's the lipid bilayer. You don't have time to draw a huge lipid bilayer, but through the magic of copy and paste, you're not impressed yet, but you will be. Ooh, there's more. And wow, right? Okay, so there's a membrane protein called rhodopsin. There's several membranes, there's several passes that the protein makes through the membrane. I'm just going to use squiggly lines like that. But deep inside, there is something which we're going to call the chromophore. This is the thing that absorbs light. And when it absorbs light, it causes a change in the shape of the protein and that allows a signal to get inside the cell. In this case, it's ions get from outside the cell to inside the cell. That causes a change in what's called membrane potential. That turns into an electrical signal in the optic nerve, and that tells your brain that you just saw light. Okay? All of that happening so fast, as we've taken forever to say it, but it's been happening while you've been looking, right? So what is the chromophore inside rhodopsin, the thing that does the absorbing of light? It turns out that it's one of the side chains of, it's a lysine side chain from rhodopsin, linked in an imine to something else. And the thing that it's linked to is a highly conjugated, oh shoot, I can't draw it from that direction. I suppose that's okay. Sorry, that's still an N when it's upside down, right? Yeah, so what's attached to that nitrogen is an aldehyde that's highly conjugated. So there's like 11 double bonds in adjacent to each other. This molecule is called retinal, and you make it from vitamin A. And you can see that it's highly conjugated. It's highly conjugated to the point that the gap between the HOMO and the LUMO of that molecule is small enough so that visible light can excite an electron from HOMO to LUMO. When you excite an electron from HOMO to LUMO, that weakens some of the pi bonds in this molecule. And that's important because one of the things you've noticed, hopefully, in this molecule that absorbs UV visible light is that cis double bond. There is steric clash in this molecule between the proton and the methyl group. That's not the most stable arrangement of the molecule. But it's locked into that configuration because of the cis double bond. When a photon of light is absorbed, that bond weakens, and it can isomerize and go to the more stable trans. Now take a look at, I'm going to draw the hydrogen that's in the trans position. Imagine what that would do for this molecule. Going from cis to trans would swing this large group from being this direction suddenly to pointing that direction, right? Dramatic change in shape. That bumps some things out of the way in the rhodopsin, and that changes the protein, allows an ion signal through, and then you get the signal that you've absorbed light. Here's the most interesting part to me is that once that isomerization occurs from cis to trans, that's called photoisomerization. Once that happens, that particular molecule of rhodopsin is spent and you can't use it anymore. Your body has a way to transport those spent molecules of rhodopsin to a different part of your retina where enzymes go in, they clip out the trans retinal, they reload it with cis retinal, and then they traffic it right back to the rod and the cone cells. And one of these, you can sort of get evidence for this happening through something called photobleaching. If you stare at a light long enough, you won't be able to see things behind that light when you look away where the light was, because you'll have an after image. And part of that's related to the fact that you've spent your molecules of rhodopsin. Okay, that was too much on imines, but we're not done yet. We'll begin with that next time. Not on Monday. Have a good 3-day weekend. I will see you next Wednesday.